She's done an extensive amount of work in related topics. She received her bachelor's and master's degree in neuroscience from McGill University, so she's more of a Montreal native, and then went off to Ottawa, at the University of Ottawa, where she received her PhD in experimental psychology. As I mentioned, she's done an extensive amount of work on cognitive training and how it impacts brain function throughout the lifespan, and work that's supported by NSERC and FRQS. And she's even the author of a book published by Oxford University Press, How Not to Train Your Brain. So today, I'm really looking forward to hearing the work that she's been doing here on the impact of genetic Alzheimer's risk on episodic memory function in healthy older adults. So I ask everyone to please mute and hold your questions till the end. And please, when you do have questions, type them into the chat box, and I will be reading them to Sheida for our mediated discussion at the end. So from now on, if everyone could just mute, and Sheida, why don't you take it away?
All right, thank you. So yeah, I'm going to be talking about some of the more recent work that I've been doing with Dr. Raja on genetic risk factors for Alzheimer's disease and how they might impact episodic memory function in healthy older adults with family history of Alzheimer's disease. So before I move forward, I just want to acknowledge all the people who have collaborated with me in the Raja Lab and also as part of the Prevent AD cohort study, and as well as all the sources of funding that have supported this work. So cognitive functions change and evolve as we age, and a little bit of a decline is expected even in healthy or normal aging. Of course, this decline becomes a lot more pronounced in conditions like Alzheimer's disease and its prodromes like mild cognitive impairment. But in the period of 10 to 20 years before any behavioral symptoms emerge, there's this a period where there are measurable changes that occur at the neural level that kind of make this a really opportune period to study the development of Alzheimer's disease and ideally to intervene and hopefully help prevent or at least delay the onset of Alzheimer's disease. And so when we're looking at sort of this period before any symptoms emerge, there is a number of risk factors that a lot of people have considered, but the ones that are perhaps the most prevalent and the ones that I'll be focusing on today are the impact of biological sex and also the ApoE gene, which is a lipid metabolism gene, and in particular the E4 allele of that gene. So if you look at biological sex, Alzheimer's disease appears to predominantly impact women compared to men. In the United States, over 65% of cases of Alzheimer's disease are in women, and even globally, there's a higher incidence, prevalence, and age-standardized death rate in women compared to men who have Alzheimer's disease. If you look at cognitive performance in men compared to women, healthy women tend to perform better than healthy men on, for example, an episodic memory task. But in Alzheimer's, there seems to be a steeper decline in women where women actually end up performing worse than men in Alzheimer's disease. And there are similar patterns when you look at hippocampal volumes. So for lower CSF amyloid beta levels and higher tau, women tend to have a steeper decline in hippocampal volume compared to men. And this sort of similar pattern when you look at the ApoE4 gene. So people who have more E4 allele, so one or both alleles being the E4 allele, tend to have smaller bilateral hippocampus and amygdala volumes. And this is sort of similar when you look at behavior. So lower scores on cognitive tests as well as memory tests. Again, in this sort of dose-dependent manner where having more copies of the E4 allele associates with worse performance. And these can be looked at as sort of additive. So again, you can see in the prevalence across age, ApoE4 having more ApoE4 alleles associates with an earlier Alzheimer's onset. But also the effect is more in women. So again, in all categories of genotype, women tend to have a higher prevalence compared to men. And so if we go back to the actual changes that are occurring across the lifespan in cognitive function. Memory, and in particular episodic memory, is one of the functions that appears most strongly affected, certainly by Alzheimer's disease. And in fact, a change in episodic memory is thought to be one of the earliest signs of Alzheimer's disease that may be able to predict the onset of clinically relevant symptoms several years before. So just a little bit of an overview on the episodic memory process. So there's different phases in episodic memory. So you can think of the encoding phase as the moment when we're forming a memory for a personal event. So, you know, being in the experience and taking in the surrounding context and, for example, the source where something is taking place and who else is around. There's a storage phase where we're maintaining and consolidating this memory over time. And finally, a retrieval phase where we're able to access these memory stores. You can kind of break down the retrieval process as a memory of just the object or event in itself, and also a recollection of both the object or event in its surrounding context. So, for example, remembering that you've seen somebody before, or that you've seen an object before, that general sense of familiarity, compared to knowing exactly where you've met somebody or where you've seen an object. And there's different theories on how this process might be activated in the brain. And so in some theories, it's thought that you first have a sense of familiarity or recognition, and then you retrieve the associating context to get the full recollection. But there's also this dual theory of memory where it's been shown empirically that you can actually dissociate the recollection of the full memory and its surrounding context from this general sense of familiarity or just recognizing an object or an event. And these are also dissociable at the neural level. So if you look specifically in the medial temporal lobes that are, of course, associated with episodic memory, it seems that more anterior regions are associated with this general sense of familiarity or object recognition, whereas posterior regions appear to be also involved in the association of contextual details. So again, if you look at regions like the hippocampus and the posterior parahippocampal cortex, for example, these associate differentially with recollection versus familiarity. And it's actually interesting, in one study, they actually were able to show that the level of confidence in a recollection or general sense of familiarity also associated with the level of activation in these respective regions. And you can look at these distinctions in regions beyond the medial temporal lobes as well. So in regions in the posterior midline, left parietal cortex, and prefrontal cortex, you can find regions that selectively or more preferentially activate for familiarity compared to recollection. And so changes in episodic memory behaviorally, when you look across the lifespan, you can distinguish, so declines in, again, source memory, so recollection versus general recognition, and source memory, so again, this recollection tends to decline at a steeper rate in all types of aging, so even in healthy aging, whereas the sense of recognition seems to be more or less preserved, or at least have a lower rate of decline in healthy aging, and this associates also with increased reaction times. But in pathology, it's not necessarily the same case. And you can, again, associate this with brain activation patterns. So for example, in this study by James and colleagues and some collaborators in our lab, they were able to show that older adults had lower source memory performance, but also that the regions that were supporting source memory performance were different in young adults and older adults. So specifically in these prefrontal and temporal regions that were supporting better performance in young adults were actually had a negative impact on memory in older adults. So these patterns, again, seem to be dissociable and correlated with behavior. In Alzheimer's disease, excuse me, or pathological aging, there does seem to be a steeper decline in recognition memory as well. So this can be seen in mild cognitive impairments, so a prodrome of Alzheimer's disease, as well as in Alzheimer's disease itself. And again, you can see these distinctions in brain activation patterns, both during the encoding and recognition phase. So for example, in this study, they showed that in Alzheimer's patients, there was a relative or less relative deactivation of regions like the posterior parietal cortex during encoding. Also, an hyperactivation of regions like the left hippocampus in prodromes like MCI. And finally, an interaction where there was a higher activation of right ventrolateral prefrontal cortex for successful versus unsuccessful trials in Alzheimer's disease. So again, you're seeing this distinction at different phases of episodic memory at different stages of Alzheimer's progression. In a study that was done by Dr. Raja and other colleagues in my lab, they were able to show that this distinction is also shown for people with different sort of degrees of risk factor. So compared to controls, so healthy controls or healthy controls who had family history of Alzheimer's disease, people who had family history as well as an ApoE4 allele showed different brain behavior correlations related to episodic memory, both during the encoding and retrieval phase. So essentially different regions that were supporting memory performance in healthy participants as well as participants with family history of Alzheimer's disease had a different, different regions were supporting memory in people with both family history and the ApoE4 allele. And what was interesting about this study, in addition, was that these changes seem to be detectable in middle adulthood. So these were all middle-aged adults in this study. So this brings us to the two studies that I ran and the two analyses that I'll be focusing on. The first one looking at the effect of ApoE4, so having an ApoE4 allele on the neural correlates of episodic memory, and in particular we wanted to know if performance and brain activity differs based on this ApoE genotype, so having an ApoE4 allele versus not having one. And also if the interactions between brain activity and behavior during the different phases of episodic memory differ between carriers and non-carriers. And the second analysis, we looked at sex differences. So again, do brain behavior relationships during episodic memory phases differ between men and women? And I'll talk a little bit about the samples as I get to the analyses, but these were, both analyses were done with the same sample of older adults with a family history of Alzheimer's disease, so specifically from the Prevent AD cohort. These were participants who were enrolled up to August 2017. So again, they all had a family history of Alzheimer's disease. All were at least 60 years of age or 55 years if their parents were diagnosed with Alzheimer's disease within 15 years, but otherwise were cognitively and generally healthy. And then we looked at specifically the task fMRI portion of the study. So this is a large cohort study with lots of outcomes that were taken and lots of really interesting results that have come out of different labs. But so I'll be focusing on the task fMRI portion. So the task itself, again, was an episodic memory task. It had both an encoding and a retrieval phase, and we were specifically looking to dissociate source recollection compared to item recognition. So this is sort of what it looked like. Participants were presented with 48 stimuli that appeared either on the left or the right side of the screen, and in the encoding phase, we're simply asked to indicate by button press if the object appeared on the left or the right side of the screen. After about 20 minutes, there was the retrieval phase that was administered. So they were shown the same 48 old stimuli as well as 48 new stimuli that were now presented in the center of the screen. And they were asked to identify with a 4-choice response whether the object was familiar, if they recalled that the object was previously on the left, if they recalled that the object was previously on the right, or if the object was new. So if you look at, or think about typical memory paradigms, there's this old new stimulus response categorization where a previously old object that's identified correctly as old is termed a hit. An old object that's incorrectly termed new is a miss. A new object that is incorrectly labeled as old is a false alarm, and a new object that's correctly identified as new is a correct rejection. But because we wanted to distinguish between recognition and source memory, this is sort of our new response, stimulus response option categorization, where an object that was correctly identified in the right source, so an old object that had previously been on the left screen, correctly identified as being on the left, was called a source hit, same thing for the right. If participants remembered that an object was old, but they made a mistake with respect to where it was on the screen, we called that a source misattribution. If instead participants could not remember where the object was presented, but they correctly remembered that it was an old object, we called that an object recognition. And if they remembered correctly that it was a new object, we call that again a correct rejection. And the mistakes were classified as false alarms if any new object was incorrectly labeled as old, either with a source or without. And any old object incorrectly labeled as new was termed a miss. And we also performed a few bias-corrected calculations. So very similar to how you would calculate D prime, so subtracting standardized false alarms from standardized hits. We calculated this probability of source and probability of recognition metric, where we looked at standardized source hits minus standardized source misattributions as a bias-corrected measure of source recall, and standardized recognition trials minus false alarms as this bias-corrected recognition. And also we excluded any participants who did not have at least 8 trials per each condition to be able to have enough power to analyze these. And the method that we used to analyze these was partial least squares. So partial least squares is a good multivariate approach to looking at whole brain patterns of activity that co-vary across time as a function of different task conditions. And we looked at task PLS for brain activation patterns during the different phases, so encoding of source versus recognition, as well as retrieval of source versus recognition. And also behavior PLS, which allows you to add a behavior vector of interest, which in our case was the performance metric itself. In PLS, significance is assessed through permutation tests. So we did 1,000 permutation tests. And essentially this gives us an indication as to whether a latent variable that arises is significantly different. And we assess the reliability of outcomes through bootstrapping. So in our case, we did 500 bootstraps. And this is just a depiction of how PLS works. So again, you kind of stack your matrix by group, then by task event, and then by individual participants. If you are doing BPLS, you add your performance metric, and then you get this cross-correlation matrix. And through singular value decomposition, you get a singular value, which sort of gives you a representation of how much correlation is accounted for by a latent variable. And then you get these brain saliences or weights that you can analyze with the corresponding design salience plots in the case of TPLS or task PLS, and correlation profiles in the case of behavioral PLS. So specifically, if you look at the upward bars, they are associated with the yellow and red regions in the brain salience plots, and the negative bar plots here are associated with the blue or colder regions in the brain weights singular image. So just to look at the results. So in our first analysis, again, looking at the differences of performance based on ApoE carrier status. So we had a relatively similar distribution of non-carriers and carriers, but of course more non-carriers. Age was slightly higher in the non-carriers. So we did account for this as a covariate in all of our subsequent analyses. But otherwise, the sample was relatively matched in years of education, as well as the estimated years to symptom onset. So this is a metric that's simply looking at the difference between the participants' current age and the age at which their parents were diagnosed with Alzheimer's disease. So again, about 10 years out from their parents' years of, their parents' years of age at diagnosis. And then we looked at neuropsychological scores. So again, more or less comparable performance on R bands, so different measures of memory and attention, but the non-carriers seem to perform a little bit less well than carriers in this case. So difference of less than a point, but it was nevertheless significant, but otherwise generally comparable at baseline. We also found comparable task performance. So again, in different categories, the performance was very similar. And if you look at our probability of source and probability of recognition metrics as well, across the age range of the participants, there did seem to be more or less comparable performance. We did notice a slight trend towards an age by ApoE interaction, where younger ApoE carriers tended to perform a little bit better than younger ApoE for non-carriers, and vice versa, but this wasn't significant. And then if you looked at response times, so again, response times were very comparable. And if we looked at sort of collapsed across both groups, the categorization of responses, we found this interesting pattern where the correct source as well as correct rejections were fastest. So people were able to most quickly respond when they were sure of an object being new or sure of what the source was, and a little bit slower for false alarms and recognition trials. So when there was a little bit of an uncertainty with respect to if an object was old or new. Looking at the task PLS, we found two significant latent variables. So the first one accounted for about 67% of the cross-block covariance, and it seemed to identify regions that distinguished between encoding versus retrieval. So specifically the positive salience regions associated with retrieval phases, and these included the bilateral claustrum, cingulate gyrus, inferior frontal gyrus, and precuneus, as well as the left middle frontal gyrus. Whereas the negative salience regions associated with encoding activity or activity at encoding, and these included left ventromedial, orbitofrontal, and anterior cingulate cortex, lateral middle, and the lateral middle temporal cortex. The second latent variable seemed to distinguish between source recollection versus item recognition. And so in this case, the negative salience regions shown again in blue here were more active during source retrieval, and these included the middle occipital, inferior parietal lobule, caudate, and temporal gyri, the left and right, the left parahippocampus and right hippocampus. And the positive salience regions were more active during recognition and associated with bilateral inferior frontal gyrus and cingulate gyrus, as well as the left middle frontal gyrus and the right inferior parietal lobule. What was interesting is that we started to see group differences when we looked at the brain behavior correlations. So up to now, the task activations were similar between groups, but when you looked at the brain behavior correlations, and particularly at the encoding of subsequently recognized items, we saw this distinction where positive salience regions were positively associated with performance in ApoE4 carriers, and these included the right thalamus, the right supramarginal gyrus, and the left insula, whereas the negative salience regions were more associated with performance in the non-carriers, and these were bilateral occipitotemporal cortices, the uncus, caudate, anterior medial prefrontal cortex, and the left parahippocampal gyrus. So this is just to show the same correlations, but maybe in a more clear way, with performance and brain scores plotted this way during the 4 phases. So you can see the difference during encoding of recognition, and to a certain extent during source retrieval. So just moving on to the next analysis. So we wanted to sort of break down the sample between men and women, but we were limited in the number of men. So the original sample had 124 women and only 41 men. So to have a more balanced analysis, we selected an age and education matched sample of women to correspond to the existing men. And the matching worked quite well. So we had a relatively similar distribution of ApoE4 carrier. We had initially intended to look at both sex and ApoE4 status together, but because of the sample, we decided not to go ahead with that, so I won't be presenting that. But otherwise, in terms of age, body mass index, and years of education, as well as, again, estimated years to symptom onset, these two groups were pretty well matched. And again, we found that neuropsychological scores were comparable between men and women. In this case, we found that women tended to perform slightly better on the MoCA than men, but again, relatively small difference, and again, both above the clinical cutoff of 26. And again, if you look at performance, so it's comparable across men and women. Here we actually found a trend for women to perform slightly better on our P-source, so our bias-corrected metric of source recall, but again, not significantly different. We did find that women typically responded faster across all response categories compared to men, except for in the false alarm category. But otherwise, again, they had the same sort of response patterns across the response categories, with the fastest responses for correct rejections and correct source, and slowest for recognition and false alarm trials. Task PLS results were very similar. So again, we found distinction in the first latent variable between encoding and retrieval that were similar across the two groups, and another latent variable that sort of distinguished source compared to recognition memory. And again, these patterns were similar across groups, perhaps less correlated in women for the distinction between source and recognition, but again, the patterns were generally the same. What was, again, interesting here was when we looked at the brain behavior correlation. So in the first latent variable of the BPLS analysis, we found a sex effect where regions that supported episodic encoding and retrieval primarily for women compared to men. These were actually all negative salience areas. So these positive bars negatively correlate with performance. These performance negatively correlated with these regions. And specifically the regions were bilateral inferior frontal, superior temporal, middle occipital and temporal gyrus, as well as the cingulate, the left fusiform gyrus, and the right medial frontal and precuneus. And again, there was no correlation identified in men in this particular latent variable. So again, just to show the correlations. And the second latent variable showed a sex by phase interaction that was particularly prevalent for source memory. So in this case, we found a negative correlation between performance and negative salience regions during source encoding in women, but this correlation was significant for source retrieval in men. So essentially showing that these regions, which included the bilateral claustrum, the left cingulate, medial and superior frontal gyrus, as well as the right anterior cingulate and superior and middle frontal gyrus, as well as the precuneus, were supporting retrieval in men compared to encoding of source recall in women. And again, just a hopefully clearer depiction of these correlations. So just to sort of sum this all up, we found that episodic memory performance was comparable across both APOE carriers and non-carriers, as well as men versus women. The pattern of response, so having the fastest response for source recall compared to recognition and false alarms being the slowest, suggests that in our case, these recognition trials might be reflective of a source failure. So in other words, participants, when they were presented with this 4-choice response option during the retrieval phase, were perhaps trying to recall the source, and if they were successful, were able to respond quickly. But if they were not able to recall the source, we're then indicating it perhaps as a familiar trial or getting a false alarm, which is reflective of these slower response times. We also saw slightly, but not significantly higher performance in APOE4 carriers and in women. So this is sort of a replication of the full sample. So at least in women, it was, we saw the same patterns in women compared to men before we had done the age matching. We also found an APOE4 status and sex differentially influenced episodic brain behavior correlations rather than task-related activity. So again, whereas both APOE4 carriers and non-carriers were sort of having the same activations during the different phases, so encoding versus retrieval, and also for source versus recognition, there is a difference in the brain behavior correlations. So we think that APOE4-based differences in the reliance on brain regions exist for the encoding of subsequently recognized objects and the retrieval of object location source associations, which may index perhaps a different approach or strategy use to the task in, again, carriers versus non-carriers. Similarly, we found activity in frontal parietal and parietal regions associated with source encoding in women, but retrieval in men. So taking this sort of together, we think that these risk factors may influence, again, the associations between performance and brain activity rather than performance or brain activity patterns alone. And so taking this further, we want to maybe look at specific regions of interest and how they might compare in men versus women, as well as APOE4 carriers versus non-carriers, and you know, looking forward in future research, looking at maybe how we can detect differences in network contributions to episodic memory. And of course, we are interested in looking at how these might interact with other risk factors, and hopefully we'll be able to look at how these patterns evolve longitudinally. So the PREVENT-AD cohort is a longitudinal cohort, although these were obviously all the baseline task analyses. So that's it, and I'm happy to discuss any questions or comments.
All right, Sheida, thank you so much for that talk. That was like extremely clear presentation on these like aspects of memory as well as partial least squares, which I'm always really excited to see. So really excellent results. So thank you so much for sharing them with us today. So if anyone has any questions, feel free to send them over in the text box. I'll go ahead and kick it off. So I was wondering if you know, you have any glimpse into what some of the longitudinal effects might actually be in these data. Have you had a chance to look at that analysis yet?
So no, unfortunately, that's part of some of the analyses I was hoping to do over the past few weeks. And of course, we're not allowed back in the facilities. But yeah, I really, we've done some very preliminary exploratory look at the longitudinal analyses, but not enough for me to be able to comment in a meaningful way.
All right, great.
That's
Okay,
the next
so we have
step.
a couple questions that have come in. One of them is, what are some of the main reasons people suspect that women have a higher prevalence of Alzheimer's disease?
Oh, that's a great question. So there's a whole literature on that I'm not so familiar with, but some of the reasons seem to relate to hormonal and reproductive history. So for example, there is evidence that there are differences in memory performance related to peri and post menopausal women compared to pre-menopausal women. There's also, you know, looking at the effect of, you know, hormonal contraceptives and hormone replacement therapy also seems to indicate an effect of estrogen on memory. So that might be one possible explanation. Yeah, I mean, I'm not extremely familiar with the literature beyond that. So in my lab, we are doing some work, not me, but my colleagues are doing some work again on hormonal history and hormone levels.
Okay. So if I could just follow up on that, do you think there's reason to believe that we might need to develop more sex-specific biomarkers when it comes to identifying like early prodromal Alzheimer's disease individuals?
Yeah, I mean, I think even in the healthy aging literature, there are evidence of sex differences in memory performance. So I think this sort of points to a bigger picture of, you know, there might be some fundamental differences in how brain activation patterns associate with memory in men versus women, you know, even in, you know, healthy aging, but you know, combined with other factors that might preferentially influence memory in women compared to men, I think this would be really an important consideration in looking at Alzheimer's development as well. So yes.
Okay. All right. So Randy McIntosh asks if you're planning to incorporate structural MRI data into these analyses.
So not in these specific analyses. There are actually a number of papers that have been published with the PREVENT-AD cohort looking at structural differences between different groups based on ApoE4 carrier status, for example, and amyloid and tau levels with PET imaging, and also just from CSF levels. So there is some published work, but at this time I don't think I'll be combining that work with the task fMRI. But maybe I'll speak to Dr. Aja about that.
Yeah, I think well, Randy's saying it'd be nice to combine these for joint prediction.
Yeah.
I would maybe also suggest it'd be great if we could incorporate structural and fMRI imaging elements into the PLS GUI in some capacity as well.
Oh, yeah. It's not complicated enough already.
That's right.
Yeah, no, that would definitely be very interesting. I mean, the nice thing is that because so many people have looked at the data in this cohort, it's comparable. So, we can take these results and look at what other studies have shown with respect to, differences or changes in, for example, hippocampal volume or cortical thickness. And so that's something that I'm hoping to be able to look at comparatively, at least, if not, within the same analysis.
Right. All right. Well, that's great. So do we have any more questions from anyone else in the room? All right. Well, Sheida, again, thank you so very much for sharing your work. I hope that you regain access to the Douglas soon. If you can go in alone at some point in time and, pull some data off the server and bring it back home. I'd love to hear more about your results as you start to examine these longitudinal studies. I hope that people will join us again for this Feindel virtual Brain in Mind seminar. Next week, we have Clara Moreau, who will be presenting from the University of Montreal. And her title is Mapping Neuropsychiatric Mutation Effects on Functional Brain Connectivity to Delineate Dimensions Contributing to Autism and Schizophrenia. So we hope to see you again, and you'll join us here at the same time and place. And please, if you're interested in presenting or if you'd like to nominate a speaker, please email me or Boris Bernhard or someone at Neuro Events, and we'd be happy to follow up with you. So thank you all very much. I hope you have a great day.
Thank you.